Muy buenos días a todos. Hoy empezamos una nueva sesión de Frontiers in Genomics y el día de hoy tenemos el gusto de tener con nosotros al doctor Purvesh Khatri. Él es investigador del Departamento de Medicina de la Universidad de Stanford en el Centro de Investigación de Informática Biomédica. Originalmente es un ingeniero en computación y se graduó en la Universidad de Sardar Patel en Gujarat, en la India, y después de eso hizo su doctorado en Estados Unidos en Wayne State University y después hizo su postdoctorado en la Universidad de Stanford y desde entonces me parece que está ahí, donde ha tenido una carrera muy exitosa. La investigación del doctor Kahtri lo ha llevado a explotar las bases de datos públicas referentes a los estudios epidemiológicos moleculares o no moleculares o médicos únicamente, referentes a diferentes problemas de salud. Y con esta ha desarrollado herramientas de estudio para analizar todos estos datos y entender mejor las enfermedades los componentes moleculares de las enfermedades que está estudiando, y con esto también, esto le ha permitido desarrollar métodos de análisis que han llevado al descubrimiento de marcas y de firmas moleculares y que le han permitido trasladar directamente estos hallazgos a la atención del paciente y obviamente esto vía la industria farmacéutica, biomédica, etcétera, para desarrollar nuevas técnicas de diagnóstico. Entonces, como les digo, su investigación es muy focalizada hacia un análisis de las bases de datos y ha tenido, con esta investigación, que ha sido, como van a ver ustedes, extremadamente interesante y bien desarrollada y muy exitosa, ha recibido varios premios de los Institutos Nacionales de Salud de los Estados Unidos y también de la Universidad de Stanford por su trabajo pionero justamente en el campo de la bioinformática. Y bueno, pues sin más, voy a agradecer al doctor Kahtri el haber aceptado la invitación que le hice. Insistí mucho para que viniera y finalmente me hizo caso. I just said that I had to write you about a couple of emails and before I could get an answer to my invitation, and I'm really, really very happy and honored that you accepted participating in our program.
Thank you. Sorry, my email is a complete mess. So it takes a couple of emails before and my apologies for that.
Don't worry, but I'm just very happy that we finally managed to contact you, and it's been really, really very enticing to listen to you.
Thank you. Should I go ahead and start?
I guess we can go.
All right. Thank you for the introduction and thank you for the invitation. It was really fun yesterday. Didn't realize that I talked for 2.5 hours. It rarely happens that I talk that much, but it was really interactive and a lot of fun. So what I'm going to talk about today is the first part is, this is more general talk. But so the first part may be a little repetitive from yesterday, and my apologies for that, but it is sort of trying to get everybody on the same page. And then I'm going to walk you through one example that would really show how publicly available data, the heterogeneity within publicly available data, and these masters that we've developed, how you can use them for diagnostics development, drug repurposing, identifying drug targets, and so on. And I'll use just one example to walk you through from the beginning to the end. So the title is Heterogeneity in Data and How It is a Blessing in Disguise for Accelerating Clinical Translational Medicine. So before we go on, I really have to thank the people who really do all the work and allow me to take the credit for their work. And yes, we have children in the lab, but we are not a child labor camp. It's just, she was like a 5th or a 6th kid that was born while the parents were in the lab. So it's a really nice group, a really smart, a very collaborative group of people. And I'm really lucky to have them to be their mentor. So For me, it started about 10, 12 years ago now, that as an electronics and communications engineer, when I started working in immunology and translational medicine, a few things didn't really, I didn't understand them. And it was in the retrospect, it was my naivete in not understanding what it takes to do translational and biomedical research. But at the time, I had this hypothesis that there is a flaw in how we do biomedical and translational research, and that's why it's slow, expensive, and all the bad things. And I'm not saying it's a mistake. Again, it's a flaw that we are in a point in time now that we have a unique opportunity to fix this flaw and really be able to accelerate translational medicine. So um, to get everybody on the same page, what is this flaw? If you think about the way we do traditionally biomedical and translational research, we usually start with a single cohort where we go out and enroll patients. We define inclusion and exclusion criteria. And the goal of these inclusion exclusion criteria is to make sure that there is as little clinical and biological heterogeneity as possible. So we try to make sure that the patients have more or less the same disease stage, the same age group, the sex bias is not there, equal number of men and women. When we generate data by profiling those samples, we make sure that we have standardized operating protocols to ensure that the technical variability is less. If data is generated in batches, then we make sure that we have a whole field in biostatistics that just deals with removing batch effects. And then if we are doing any kind of biomarker discovery or trying to understand what the pathways that may be, the way we do analysis is we divide by dividing this cohort into sort of training and test, or discovery and validation, where we only use the discovery cohort samples. We ensure that we do all our repetitive analysis in those samples. The test samples are never seen by the iterative process of identifying biomarker. So overall, the way to think about this is, when we only use blue people and then try to validate in blue people, which is the left out sample, and we can show that everything else being equal, this biomarker or this pathway does show up in another similarly blue people, and that becomes a paper. And then it never immediately goes in clinical practice, because we know that this single cohort does not capture the real world patient population heterogeneity. A patient with infection could be coming from any country in the world. They may be children or adult or older people. They may have different host genetic backgrounds and so on. So results don't generalize. And, and that's sort of these green people, that what we identify by analyzing only blue people, it does not translate to green people. And this is what leads to the translational medicine, what's called valley of death, where a lot of the things that are discovered in the lab or by the bench, they don't translate to patients' bedside. Now, in statistical terms, what's going on here is, if you think about, you have to think Bayesian prior. So if when we generate data, let's say RNA-seq, single cell or bulk, cytometry, proteomic data, it doesn't matter. Pick your favorite technology that you are interested in. Before we analyze, or before we select a set of variables, each variable has 50-50 odds in favor of being associated with that condition. Then we do our regular analysis, let's say p-value 0.05. And then when we try to replicate, what we don't realize is that there is still about 30% chance that something that had an odds of 50-50 odds of being associated with condition, it would still not replicate. And this is where a lot of the data reproducibility crisis discussions are coming from. But we aren't really talking about this Bayesian prior, that we are always selecting from a set of variables that have 50-50 odds in favor. But if we simply change one thing in upfront, before we do replication, before we select a set of variables for replication, if we simply select from a set of variables that already we know have 90% odds of being associated with that condition, then select from that pool with a p-value 0.05, we have less than 5% chance of it not replicating. And the statistical background about this whole Bayesian framework is in Steve Goodman's paper now 20 years ago. It's a very short paper, but highly worth it. I highly recommend that you read that paper. So how can we go from 50-50 odds to 90-10 odds in favor? And that's where we have to start embracing the heterogeneity. And that's where publicly available data becomes really useful, because these publicly available data sets, they come from different labs in different countries that looked at different patient population using different technology. So all the confounding factors that we traditionally try to control in biomedical and translational research, across these data sets that are publicly available, they are all present collectively. So now, if we can somehow combine all of these data sets, or even a subset of these data sets, in a single analysis, so that now we know that there are some data sets that come from blue people and some come from green people, some come from children, some come from adults, some come from older adults, then not only we know that it will work in all of those people, but it also has a higher probability that it will work in patient population that we have not yet analyzed or observed. And that would be like an orange or a purple people. So that's the intuition behind it. By combining a lot of heterogeneous data, we are going to identify a set of variables that have 90% odds in favor of being associated with that condition, which then increases its probability of replicating in other patient population. So we'll find more generalizable solutions. But then the question becomes, this is so heterogeneous, it's really dirty data. We've been talking about all of these data not being reproducible. So how do we really integrate this data? And that's the research that my lab has been focused on for almost a decade now. So, if you remember nothing else from today, this is the slide that you should remember. This is a distillation of last 10 years of our experience and everything into a single figure, where we start everything that I'll show you today. Remember that it all started with publicly available data. I'll point out which data we generated versus how much data was publicly available. And that's another way to think about in your own research is how we are using publicly available data to generate hypotheses and our own samples to validate those hypotheses. So we are not wasting our own private samples to find new hypotheses, but to validate those hypotheses. So we are much faster. We already have a validation cohort to show that when everything else is controlled, that conclusion still holds and it is not biased by anything. So we always start with publicly available data. We make sure these data sets, once we pick a disease that we are interested in, we go in public data repositories like Gene Expression Omnibus, Array Express, GTEx. The list is just very large. From that, once we've identified data sets, we download them and we make sure that they are normalized within themselves. We never normalize between data sets. And even when we do slightly different. We only normalize the healthy controls. We never normalize healthy controls and the disease samples across all data sets together, because normalization in a way is itself a kind of a confounding factor. Once you've identified these data sets, we divide them into discovery and validation, just like we do in a traditional research. However, we never divide any data set into both discovery and validation. So an entire data set either is in discovery or in validation. And when we decide which data set in discovery and validation, there are two guiding principles. The first principle is if there is one group that has multiple data sets in public domain, then all data from that group is either in discovery or validation. So we never allow the same group to be in both discovery and validation, because we really want to ensure that when we validate our data, our results, our models, it is truly coming from a completely independent group And the second guiding principle is we choose data sets in discovery such that they are as heterogeneous and as different from each other as possible. So we try to ensure that these data come from they are profiled using different platforms, they come from different countries, different patient groups, like the age or a different stage of disease and so on. And we try to add all of those in our discovery. And the idea is to allow our discovery process to see as much heterogeneity as possible and find a generalizable solution as generalizable a solution as we can find. How much data sets do you need? You need about 4 to 5 data sets, and all the details are in this nucleic acid research paper. And I won't go into those details, but you need about 4 to 5 data sets, roughly 200 to 250 samples total that include your control samples. So you don't need 200 samples from your disease group. As long as you also have a similar number, half and half of controls and cases, you are good to go. All of our source code and software code is publicly available. So Metaintegrator is where you use that to analyze data. And the tutorial and all of that is also available. But if anybody has questions, just send it to me and somebody in the lab would answer. And how do you select genes? These are, again, keep in mind, these are all guidelines. These are not rules. There is no penalty for breaking the rule. Generally in the lab, we use FDR of 20% because the way this analysis is done, we are very stringent. So FDR 20% is fine. And we've repeatedly found results that have replicated over and over and over again. And what this left-hand side of the framework does, it identifies a set of variables that have 90% odds in favor of being associated with that condition. That's what it does. Now you can use these variables, apply your favorite machine learning algorithm if you are looking to develop biomarkers and a classification or a prediction models, or you can use these variables to see if they are telling you any new mechanisms, any new therapies, or can the classification or prediction models be diagnostic and prognostic. But this is done in data sets that come from completely independent groups. So they are generated completely independent of the data sets that were used in a discovery. And the question I always get asked is, would this really work? And just to put most of our results on a single slide and to convince you that this really is a generalizable framework that works across a broad spectrum of diseases, we've been able to show that, so we've just applied it to a large number of diseases that include organ transplant, diagnosis of sepsis, infectious diseases, predicting treatment response in autoimmune diseases, predicting vaccine response before somebody gets vaccinated, and so on. And what you need to remember on this slide is a few things. Number one, all of the data was publicly available. We did not generate a single data point. This was all publicly available from NCBI Gene Expression Omnibus. Number two, each of these study was done using only human clinical samples. So there was no model organism or anything like that. It's all real patient data. Number of data sets range from 7 to 40. Number of samples range from something like 900 or 700 to 7,500. And each one of those, we were looking to develop a diagnostic. And we found a set of genes, we built a diagnostic, we validated in unseen data, and then when we designed our own trials to further prospectively validate it, we were able to show that each one of these gene signature actually is predictive, meaning it predicts that corresponding clinical condition before a clinician can diagnose that. So, for example, we could predict who has subclinical transplant injury going on 18 months before a clinician could see it on a biopsy. We could diagnose who has sepsis about 24 to 1 to 5 days prior. This is my favorite example where people were challenged with influenza, wild type influenza. And you see this one red that is asymptomatic but looks like symptomatic, and this one blue that's symptomatic but looks like it's not infected. So we actually talked to the original study author, and the study author told us that this person, this asymptomatic that looks as if is infected or symptomatic, was somebody who shedded virus every single day after the challenge but never showed symptom. So our signature was actually saying, although you are not showing symptom, you are asymptomatically infected. And this person who showed who showed all the symptoms but never shedded virus over 7 days, our signature said it's not infected. So that's the specificity and sensitivity. Out of 4,500 samples that we analyzed, 2 samples that were incorrectly labeled in terms of infection status, not the symptom status, but infection status, we were able to pick them up. So that's the sensitivity and specificity you can get. All right. And how do you learn a new biology? So while we've been really focused on developing these diagnostics for infectious diseases, what we've actually learned is that immune system is really a very it has its own hierarchy, where if somebody shows up with, if somebody has acute illness, we can actually say whether they are infected or not, so infected or not infected, what kind of pathogen they have, and in many cases, what the actual bug is, as in, is it a Mycobacterium tuberculosis, influenza, dengue, malaria, and so on. Every gene signature that you see here in these black circles has been prospectively validated, and 5 of those signatures, the ones highlighted in red in the bottom, have now been translated on a point of care device and are moving towards commercialization and regulatory approval and so on. So that's the speed, right? We could go from describing these signatures in last 6 years, 2015 to 2019, and 5 of them are on their way to commercialization. So you can imagine how a small group of less than 20 people was able to do this in 5 years across a large number of, across multiple different infectious diseases using only publicly available data. So I'm going to walk you through one example to really show you that you can really do all the things that you'd want to do: diagnosis, prognosis, finding disease mechanism, finding drug targets, drug repurposing, and so on. This paper just got accepted in Immunity, so it should come out soon. And these 7 people came together when the pandemic, COVID-19 pandemic hit, and said we should do something where the results are more generalizable. And really, they started on May 13th and submitted, as you'll see, a fantastic piece of work in like 3 months. So what did we hypothesize? So COVID-19 is a 4th pandemic in the last 10 years. We've had H1N1, Zika, chikungunya, SARS-CoV-2, Ebola was near pandemic, but it was never declared officially a pandemic. So we are going to continuously keep having these these viruses emerging and spreading really fast across the world. And each time we cannot go back to drawing board, trying to find a solution that only works for that virus. And then the next time we have to go back to square one every single time. We need solutions that are more generalizable. So 5 years ago, we had published this paper in Immunity where, where we had shown that there is a conserved host response to viral infection that is actually different from bacterial infection. And it was a 396 gene blood-based signature. So we hypothesized that while we had identified these gene signature to be common across all the patient populations across the world, irrespective of age and so on, that signature, those 400 gene signatures, would also allow us to identify patient subgroups to say who has or will have a severe outcome versus who's going to have a mild outcome. So now a conserved gene signature to identify patient subgroups to predict or diagnose what's happening to them or what's going to happen to them. So, sorry. Right. Okay. Sorry. So to test this hypothesis, what we did was, when I say we, it's a royal we. It's the 7 people in the lab. They scoured Gene Expression Omnibus and Array Express, found that there were about 26 data sets that had 34 independent cohorts from 18 countries where the patients had one of a viral had infection of one of the 16 viruses, and that was about almost 4,800 samples. And while we were collecting these data and curating and so on, there were 3 single-cell RNA-seq data sets from 3 different centers in the US of patients with SARS-CoV-2 infection that were also available. And that was about another 300 samples, 700,000 single-cell RNA-seq data. So we use these data sets to now test our hypothesis. And keep in mind that we are not doing any more discovery here. Here we are simply testing a hypothesis that those 400 genes can also tell us which patients will have severe outcome versus will have a mild outcome. So that's why they are divided into discovery and validation, to sort of ask new questions using only those 400 genes and then test them. And now each of these samples, all of these 51, 5,200 samples, we then assign them one category, one of these 7 categories. Are they healthy or asymptomatically infected or convalescent, mild, meaning they recovered at home, or they came to hospital, but or the emergency room, they were admitted to hospital, but did not have supplemental oxygen, no mechanical ventilation, no ICU, so they would be moderate. And if they are in ICU on mechanical ventilation or needed supplemental oxygen, they'll be severe, serious, critical, or fatal. And once we had this, the first thing we were able to show is that if we just look at the score that we defined 5 years ago, and it is logged using this metavirus signature, it correlates with severity. Correlation is 0.75, really high correlation. It does separate healthy from all the other infected patient population. And it doesn't really differentiate between mild versus moderate. So that's so far so good. When we look in data generated using RNA-seq for viruses that were either pandemic or near pandemic, we still see the same correlation. Now, keep in mind that the SARS-CoV-2 patients, their samples were collected at the time of presentation before the outcome was known. So the host response is actually correlating with patient severity before the outcome is known. The same thing for the Ebola patients, where the samples were collected at the time of diagnosis, and then outcome was known in the next 30 days. So again, this is really robust. This data is done profile using RNA-seq. So we are still seeing the conserved host response validating in a completely unseen viruses that the data wasn't available 5 years ago. But what was even more interesting was in this RNA-seq data, we could actually detect viral RNA read. So like RNAmia, not necessarily viremia that the viral particles are in blood, but at least the RNA from the virus is still in blood samples. And we could show that host response on y-axis actually correlates with how much viral RNA is detected in peripheral blood. So host response is keeping up with viral load in some sense. Then the second questions we asked was, now that we know this host response is conserved even across the new viruses that are emerging, where is this conserved host response is coming from? Because immune systems definitely conserving. So we took these three single cell RNA-seq data sets, over 700,000 single cells from PBMC, where the Blish lab data was using sequel versus the other two were done using CiteSeq, so different single cell RNA-seq technologies. And after we aligned the three data sets, and we could show that it basically clusters by the immune cell type and not by center and so on. When you color each cell by the MVS score at a single cell level, you see that it's mostly coming from myeloid cells. And the cells that have the highest MVS score are those coming from the patients with severe outcomes. So now you can see that the gene signature that we identified using bulk transcriptome, it actually is reflected at a single cell level in myeloid cells in patients with severe outcome. So now this is how you can see, using publicly available data, we are starting to do systems immunology. The next question was, when you think about gene expression in a bulk, there are two questions, right? One question is, when we see differential expression, we don't know whether that differential expression is because the number of transcripts per cell are changing, or just the number of cells are changing without changing the transcriptional program inside the cell. So we've developed a method for being able to figure out what the cellular proportion of 20 different immune cell types are using the bulk transcriptome profile. And what we were able to show is, number one, if you just look at the single cell RNA-seq data, we see that the MVS score at a single cell level in myeloid cell continues to increase with severity, but that increase is more in CD14 cells, not in CD16 positive monocytes. So that is at just the transcriptome. And then the question you can ask is, but what happens to the cell proportion using single cell RNA-seq data? And we could show that, yes, as the severity increases, the proportion of myeloid cells also increase, but that increase is driven by CD14 positive monocytes, not CD16 positive monocytes. They actually go down with severity. And then the question becomes, would we see this in a bulk data? So using our method called immunostates, now we went back to the bulk data that we had already collected, and I showed you earlier. And now we deconvolve those to estimate different immune cell proportions, the 20 different immune cell proportions. And we found that in non-severe patients compared to healthy, total monocytes do go up, but all of that increase is driven by CD14 positive monocytes, not CD16 positive monocytes, just like you see at a single cell level. And in severe patients, so those who are in ICU, mechanical ventilation, and so on, compared to healthy controls, you don't see total monocytes changing because CD14 positive monocytes go up and CD16 positive monocytes go down, just like we see it at a single cell level. So now on this figure, what we had observed using bulk transcriptome data 5 years ago, now we are seeing where that signal is coming from, that not only the transcriptome is changing at a single cell level, the proportion is also changing at single cell level. And then we are validating that observation across all the other patient population that we already have bulk data. So now we can go across data sets, single cell or bulk, cell population or transcription profile, all of that using just publicly available data. And we can start asking these questions that we would ask in a lab, but now we are asking it across the entire patient population. And here you have data coming from children, adults, adolescents, so you can also appreciate how robust these signals are. Now, the third question we can do, and please, if I run out of time or something, please, Yvonne, let me know. But the next question we asked is, just like in a single cell RNA-seq data, we have each patient is our, each cell is a snapshot along the cellular differentiation trajectory. So now we can think about patient, each sample being a snapshot of the patient's disease trajectory. So we used a subset of samples, 1,700 or so samples. Using those 400 genes, we built a UMAP, and we could show that healthy samples are on one side and viral samples are on the other side. UMAP first dimension just captures the severity of the viral or just the MVS score. And mild samples are on one side, severe are on the other. And when we map another 8 studies, 7 challenge studies using influenza, rhinovirus, or RSV, where samples were taken every 8 hours, the one that I showed you earlier where we could identify symptomatic or asymptomatic, where nobody progresses to have severe viral infection, none of their samples, except this one sample, go in the severe space. And SARS-CoV-2 patients, where we don't know the outcome yet, but we when the samples were collected, but those who end up having severe outcome, like either they die or have acute respiratory distress syndrome, they already start out in this space where the other severe patients are. So it's telling us that it's really robust. So now we said, just like we do these cellular differentiation trajectories in single cell RNA-seq data, can we do that in patient samples? So this method, another postdoc in the lab, Dennis Dermadi, has created this method called T-space. So we modified it slightly, very slightly. Still at its core, it's still T-space, but we call it a D-space, as in disease space. We applied it to say, can we identify what happens to the patient's disease trajectory? So in this disease space, when you do principal component analysis, you see that principal component 1 just captures the severity. So going from right to left, it's healthy, asymptomatic, mild, and then the severe patients. But principal component 2, it's just a straight horizontal line separates mild versus the severe patients. So we applied phenographs just like we do in a single cell RNA-seq data. We found that there are 10 patient clusters. And if you look at these clusters and the proportions of patients samples with different severity, sample 1 through 5, cluster 1 through 5, it's just healthy or convalescent or patients with asymptomatic viral infection, more than 80% of samples. Cluster 6 through 10, it's just mild samples. Cluster 13 through 20, as you go from 13 to 20, proportion of patients with severe outcome continues to increase, and 11 and 12 are the most heterogeneous. Every category is present there. So now we can build a trajectory that goes through these clusters. So that's what you see in the lower right panel, panel D, where on one side we have severe patients, healthy are in the middle, and then the patients with mild outcome are on the other side of the trajectory. And now you can say, what are the genes that change along this trajectory significantly? And they will be the ones that are really interesting. So when you do that, and then the disease space allows you to do that, we identified 96 genes. So what you see here is at the top, you have, so that V-shaped trajectory is now linearized. So on the left-hand side, you see severe patients, mild patient, healthy in the middle, and patients with mild outcome on the other side. Each column is one patient. So you are looking at 3,200 samples here. And each gene's expression across all 3,200 samples along that trajectory. And then the circle heat map is another 6,600 samples from sorted immune cell types, and then the bulk transcriptome to say where are these genes, which immune cell type these genes are coming from. And then this last one is the correlation of that gene's expression with the severity. So you can see these first three, especially cluster 1 and 2, the first two are positively correlated. They mostly come from myeloid cells, and they are positively correlated with severity. And the bottom one, the blue cluster, that's negatively correlated, and most of the genes come from adaptive immune systems, T cells and NK cells, and so on. So now how do you use this information? Now you can say, look, I see that some of the genes are highly expressed in hematopoietic progenitor cells. They are very specific. So I think that, so the hypothesis we tested was that hematopoietic progenitor cells increase with severity. So first we could show, so here on this figure, you would see how we are using, it's basically a systems immunology using only publicly available data, where first we show, we confirm using the same data that indeed in those 3,200 samples, as the severity increases, expression of these genes also increase. It validates in a completely unseen data. So SARS-CoV-2 data set is always used as validation, and indeed that happens. In single-cell RNA-seq data from those three data sets, as the severity increases, proportions of HPSCs, hematopoietic progenitor cells, also increase. And you see it's very small proportion, like less than 2% usually. And in bulk transcriptome data, where a lot of these samples are whole blood, so the proportion is even less than 1% of HSPCs, we are still able to detect a small change in patients with severe outcome compared to healthy or non-severe outcome. So that's the sensitivity that immunostate gives you to even detect the slightest change in the rarest cell population in bulk data across the large patient population from many different countries and so on. So here you can again see how using publicly available data, we can leverage heterogeneity to do systems immunology. Another observation was a lot of the markers of myeloid-derived immunosuppression were also changing with severity. They were positively correlated. We can see that indeed we see the same thing in COVID-19 patients. And when we do deconvolution, we see that in non-severe patient compared to healthy, pro-inflammatory macrophages go up, anti-inflammatory macrophages go down, so immune system's doing what it's supposed to do. But in severe patients with severe outcome, not only pro-inflammatory macrophages go up, anti-inflammatory macrophages also go up. So somehow there is immunosuppression coming along. Again, a very conserved thing. Same thing we see in NK cells. Markers of NK cells go down. We see NK cell proportion goes down at a single cell level, and also the same thing in a bulk level as well. So now what else can we do? The next thing we can do is we can ask a question of, because these genes are highly correlated with each other, we don't need all 96 genes. We can just look at a subset of genes, like the genes that mostly highly differentially changing along the trajectory. So we found 42 genes. These 42 genes belong to one of these 4 clusters that are colored on the dendrogram on the far left. And when you just do a geometry, when you define a score, and we call it, it's just a geometric mean of all the genes in each of those clusters, in each of those modules, and you can see those genes on the modules in the panel B over here. Overall, the module scores are also positively correlated with severity in module 1 and 2. Module 3 is very interesting. Its expression is really high in mild, but in those with severe outcome, it's not as high as in mild. And module 4, the module's 4 score is in mild is not different from healthy people, so it's just there. And in as the severity increases, module 4 starts to disappear. And remember, they come from these adaptive immune cells. The next thing you can ask is, how do the correlation between these genes themselves change in healthy, mild, and moderate patients? And you see that this column for the healthy is like the basal correlation structure in PBMC samples between those genes, and you see that there is some correlation. And you see the distribution of these pairwise correlation values in the far right. So compared to healthy, module 1 and 2, they don't go up. Like module 1 score is just as it's in healthy people. In module 2 score in mild, it actually goes down, and this is like the anti-inflammatory macrophages that I showed you, that they go down, and you see the same thing here. Those are the genes that are biased towards myeloid-derived suppressor cells. Module and in severe patients, module score 1 and 2 are just going through the roof. They are all going together. Module 3 is really interesting because they only correlate with each other in mild patients, not in patients with severe outcome. When you look at that distribution of correlation structure, there is like no change. So module 3 is my favorite. So we looked at it more, but before, so now how can you use this in diagnostics? So we defined a very simple score. Module score 1 and 2, which are positively correlated with severity. And so that's the sum on the numerator, and the denominator is module 3 and 4 score. And you can see that it very nicely separates those with severe outcome from mild outcome, and healthy are in the middle. What was even more interesting is this module 3 and 4, if you look at it in nasal swab. So a data, while we were, just after we submitted the paper, a nasal swab data from UCSF became public. And we could show that the protective modules, not the detrimental modules, module 1 and 2, they don't show up in nasal swab because they are more myeloid-derived and more specific to peripheral blood. But module 3 and 4 show up in exactly the same direction as we see in peripheral blood at the site of infection. They are also negatively correlated with severity, very significant. And as the severity increases, these modules are not present in a nasal swab as well. So then 42 genes, you can argue that they are very difficult to translate clinically. Can you even reduce it further? So we've even reduced it further. This paper is under review right now. But now it's only 6 genes, and you see the 6 genes in panel B, where 3 genes are upregulated in patients with severe outcome, and 3 genes are downregulated in patients with severe outcome compared to those with non-severe outcome. And in 2 completely independent cohort, prospectively collected samples, where we don't yet know the outcome, we could show that a locked model using publicly available data actually predicts or diagnoses which patients have severe outcome or will have severe outcome, and who have mild outcome or will have mild outcome. Remember, this GRIS cohort is completely prospective. We didn't know the outcome when we collected this data. And it's very high specificity and sensitivity when you look at the AUROC curves. And now we have translated this into a LAMP-based PCR assay, and we are able to show that the LAMP-based PCR assay gives exactly the same results, highly correlated results, as if we were to measure the same sample using a nanostring. And we are submitting this for FDA emergency use authorization in order to be able to triage patients when somebody gets diagnosed with a viral infection. Right now for COVID, but in general, this should work for all the viral infections in the long term. So what I just showed you is using a signature that we had derived 5 years ago using large amount of publicly available heterogeneous data. We then expanded it to another 5,000 samples and using integrated with single cell RNA-seq data set to show where it is coming from. Then we built a patient trajectory to show that here are the 96 genes that are different between patients who progress to a severe outcome versus who have mild outcome, that also tells us what are the different, how the immune system is changing across cohorts, irrespective of age, pathogen, or the pathogen strain, and is conserved, that can then be used in a clinic for predicting severity. And we did all of this from May 19th through the early November, so something like 6 months, right? And now in just 2 slides, how can you now use the same signature, same metavirus signature, to say, you know, we have a problem with antiviral drugs that each antiviral only works for one virus and it doesn't work for multiple viruses. If there is a drug that works across multiple viruses, because viruses need host replication machinery to replicate themselves. So if we can find a conserved host pathway that multiple viruses need and target that, then we can have a broad spectrum host directed antiviral therapy. And the viruses will have difficult time mutating against that, because this is the host factor that they evolved to use in their life cycle. So we've developed another method where we can now use a large number of compound screening studies from PubChem, another publicly available data, use a subset of those, then integrate that with our host response, conserved host response signature, the same one that I just showed you, and transcriptional profile of over 20,000 small molecules, and ask a question: find me all the drugs or all small molecules that look like these bait compounds and also look like the conserved host response. And we found, and we predicted drug, and first we validated it in vitro. Here is one example, BCL2 inhibitor, that first in a dengue, because we had not analyzed dengue anywhere in all of our data. So we directly went to dengue to say, if our hypothesis is really true, our predicted drug should work actually in a completely unseen virus. So first in vitro, dengue infected cell, it kills the infected cell, but does not kill uninfected cell, meaning it is not cytotoxic. In vivo, dengue infected mice with lethal dose, after 72 hours of treatment with this molecule, it reduces the viral load. The mice survive. We've repeated this experiment now 4 times. It's true. And the mice that survive after 2 weeks, they all regain their weight and so on. And what is really interesting is this drug has been through now 17 or 19 phase 2 trials, about 700 patients. Patients have been treated with this drug for like 2 years and no side effect. And it doesn't cure cancer after 2 years, but 7 to 10 days of treatment might actually cure viral infection, and it might work across the broad spectrum of viruses. So I'll stop here by just summarizing that heterogeneity in data is a blessing in disguise. And we need to rethink how we do biomedical translational research, where we start accounting for biological, clinical, and technical heterogeneity upfront to identify generalizable solutions, which we can then use to also do patient subgroup identification, drug repurposing, and so on. All of the methods that we have talked about, we have a large number of methods that when you can use, and I showed you one example where you can use it in a single analysis to really do an in silico systems immunology analysis to go from hypothesis to a clinically usable assay in less than 9 months, as well as a predicted drug treatment. I'll stop here by thanking again the people who do all the work, investigators who share data, collaborators who do all the trials for us, and the funders. Thank you. I'll take questions now.
Right. Thank you so much. It was very interesting and very important nowadays with all what we are going through in COVID and so on. So I think that was really very interesting to find out that you can now analyze all those new data. And so yeah, I say that we could ask questions. Okay, so there's one. You want to read them? Because it's no
Okay,
need that
so
I...
there is a question from Elsa. How often are these gene signatures translated efficiently to the clinical setting? What are we lacking to ensure that research effort like yours don't get lost in translation from the lab to the clinic? And so that's a great question. And I can say for myself that in the last 6 years, we since we started publishing these gene signatures, we, in the next 2 years or so, so total about 7 years, we are on track to have 5 diagnostic signatures in infectious diseases on the market. So how can we do that? There are a couple of things. One, we need to fundamentally change the attitude in academia that things identified using retrospective data are just some bioinformatics mumbo jumbo. We really need to appreciate how much data that has been used, how robust the analysis is, and ultimately don't look at it as data reanalysis or being data parasite and all of those things, but how it is useful and how it can drive the field forward. So that's number one. And number two, um, uh, really, and then the reason I say this is because when we publish these and we try to license to the companies that exist, they all went away. So ultimately, Tim Sweeney, postdoc in my lab at the time, I and one of our other co-founder, we just decided that now we have to just go ahead and do it. So we started a company and just decided to do this the same way. So that's the answer. If nobody else does it, and if you believe in your data, just go ahead and do it yourself and show that it's true and you are not hallucinating. There is another question from Esperanza Martinez Romero. Do transcriptome data match proteome data? Great question. It depends on which part of the transcriptome data you look at. So if you look at, in my experience so far, I don't want to make a very general statement, but in my experience, anytime our Metaintegrator framework said by analyzing across multiple data sets that the effect size of this gene is greater than 0.8, which is in a log2 scale, and then we tried to do proteomic analysis, that protein has always showed up. So I think it's a function of the fold change. What is the effect size across multiple data set? And based on that, if it's a high enough effect size, the proteomic side also tends to show up. So far, I haven't found a protein that did not show up after we predicted on a transcriptomic side, but there is always first time to be wrong. So that would be the answer. It's a cop out, but I think that's an honest answer. Hiromi has a question that I've read in the literature that there are sex differences that may determine the fate of vaccination studies or other treatment strategies, particularly hormonal variances. So the question is, and in a way, main interest is, how do these approaches are studied regarding trans people and their hormonal transition path? Are there any efforts to approach this situation? That's a great question, and I wish there were more data sets available to answer this question. But what I can tell you that we have done in terms of sex differences is we asked exactly the same question. And the paper is published in Cell Reports. The first author is Erika Bongan, where we actually showed that we didn't look at any disease sample, only healthy men and women across something like 12 countries, over 5,000 samples, and showed that indeed, if you just look at men versus women, just healthy, and 18 to 40 years old, the genes that are autosomal genes, so you see, of course, see X and Y chromosome genes, but autosomal genes that are really different between men and women across every culture, every genetic background, it's always there. And just by using those genes, we can take any sample and with an AUROC of 0.8 tell you whether it comes from a man or a woman. So that's number one. Number two, there were 2 data sets or maybe 3 data sets that were from like Klinefelter syndrome, people with Klinefelter syndrome, right? So extra X chromosome, and there was one data set, both of them had extra X chromosome, but also had Y chromosome, right? So if you look at those samples, or samples from people with Klinefelter, so extra X chromosome, what you see is really interesting. And this is a main figure in the paper as well as a supplementary figure. If you look at even just the X and Y genes, you see that the score that we defined, so it would be higher in female and lower in male, just the X and Y genes, just looking at X and Y genes, the Klinefelter is right smack in the middle. So you actually see a trend where you go from women, XX women to XXY to XY men. And the same thing happens with autosomal genes as well. So that's another way of thinking about it, that maybe we don't have more data on trans people, but maybe we can start thinking about, and it may not be the effect of hormone, and it may be the chromosomal dosage effect, but these are a few ways to start thinking about how we can do it. There is one more question from Diego Camacho. So you derive a specific viral signal from metavirus signal in a top to bottom fashion. Can we expect a specific subset of genes for every single infection disease? Is this possible from an evolutionary landscape? That's my hypothesis. And we are going systematically to see if we can see this. Now, what we have found so far, and maybe it's a testable hypothesis, but we need more data from different viruses and pathogens to be able to do that. So what I can tell you, what we have found is influenza has a very distinct host response from RSV and HRV, and but not from dengue as much. But it also has this conserved viral response. And if you look at in challenge studies, where you have longitudinal data, what you see is the conserved viral response comes up first, and there is always a lag by patient, where the influenza response turns on or comes up about a few hours later. It depends on different people at different time point. So what that tells you is like immune system is making a decision. Is this a bacterial infection or a viral? If it is a viral infection, it turns on this innate response to viruses and then says, what is this virus? And then it turns on this influenza specific response. We've done the same thing in TB, where we are able to show that we can diagnose TB infection compared to all the other bacterial pneumonia patients. We can also distinguish intracellular bacterial infection from extracellular bacterial infection. So there is definitely this thread, and it's like immune system has learned evolutionarily, but we just need to keep testing this hypothesis. I think that's it.
So I have a question, a very fast question. Probably it's not possible, but this kind of correlation analysis can be also predictive for the capacity of the immune system to generate a strong immune response against a new infection. For instance, you can predict what's going to happen depending of the different patients. For instance, in the case of COVID-19, by the expression analysis and the correlations that you perform, can you also eventually predict what people or which person is going to have a better immune response for a future infection? That's possible?
So are you saying, can we predict who will get infected upon exposure while they are healthy?
Yes.
Yes, we've actually done that, where we showed that we could predict by analyzing healthy people who will get infected or who will remain uninfected if they were to be exposed to influenza. And then we validated in an influenza challenge study to show, and it was a single gene marker. It basically comes down to NK cells. So if your KLRD1 expressing NK cells are lower, then you are at a higher risk of being infected upon exposure. And if your KLRD1 expressing NK cells are higher, then you are at a lower risk of being infected if you were to be exposed to influenza. And I think it's going to be more generalizable because NK cells seems to be showing up across several different viral infections. They are also associated with the severity and so on. So I think we need to do more tests. But yes, so the answer to your question is yes. We've actually done it, and I think it can be done.
Okay.
There is a question from Isaac. Maria. Is there any minimum number in the sample size, particularly of the controls? Actually, no. So yes, what you need is, and then I'll recommend that you read our nucleic acid research paper. But you need about 4 to 5 data sets, total 200 to 250 samples. But most importantly, the cases and controls should be roughly equal. So don't have 10 controls and 90 cases. Each data set should have a moderate statistical power and roughly the same number of samples. Not exactly, but roughly the same number of samples. And you'd be good to go.
Muted. I guess we're getting to the end. And well, we wanted to thank you so much for this. This has been very, very interesting. This is now the third time I have the privilege to listen to you, and it's always very interesting. So
now you are bored.
Now
you must be bored of listening.
I'm not bored at all. It's well beyond what I do, but I think it's really very interesting and very suggestive of future things to do and things to come. So, well, thank you so much, and we'll keep in touch, I guess.
Definitely, and hopefully the pandemic would be gone, and I'll get to visit.
And then you can come to Mexico, exactly.
Yes,
that will be better. It's easier when it works that way. It's easier and it's nicer too, because we get to have a real closer contact with our guests.
Definitely.
Okay.
All right.
Well, thank you so much.
Thank you for the invitation.
No, you're welcome.
Bye.
Thank you so much. Bye-bye.